We'll have to wait for a couple of minutes for everybody to arrive. Although this might be it today. We'll just wait another minute.
That wasn't a French, but what I was doing. Like every single degree I got, every single degree I had was it's outline quickly. And that's it. You spoke about it. It's interesting what he says.
Right. Okay. Well, I think this is it. So, no worries. So let's talk about gas exchange. God, it feels like a long time since we did this. Hello, Fran. Come on in. Oh, I shouldn't be using your name. I'm recording. Hello, student. Right. Okay, let's get started. Exchange. So first question then is, what do we exchange? What do animals exchange? Sorry? What do we exchange? Yeah, oxygen, CO2. What about in the plasma of our blood? What else is dissolved in there? Yeah, urea. Also, we need to, rather than exchange, we need to ensure that we keep a certain temperature, same temperature, so homeostasis and things like that. And there are certain adaptations of different types of animals as well. So we'll talk a bit about that. So let's talk about surface area to volume ratios. So first of all, okay, so we're going to do surface area to volume ratio. So what kind of animals would you expect to find, what in terms of their surface area to volume ratios if they lived in cold environments? Sorry? Smaller surface area to volume ratio? Can you tell me, so what would you expect to see in terms of adaptations? Well, they tend to have small ears. They tend to have a round head. Those small ears, round head, small tail, and all of that is to, it depends on the individual example, but all of that is to decrease the surface area to volume ratio. So decreases it, okay? So it reduces heat loss, particularly in that environment, okay? To reduce heat loss. Okay, conversely, in a hot environment, what kind of features? Yeah. Big ears, larger, longer ears, yeah. Usually thin legs, tails, like rats and mice and stuff like that. Pointed noses. So that increases the surface area to volume ratio. Okay. So that will increase heat loss in particular. Okay, you might be expected to calculate surface area to volume ratios. So calculate. So they'll do a 1 to something ratio usually. So so surface area, it depends on the shape, but usually surface area is, does anyone know what the calculation is? So it's 6a squared, basically. Is that correct? Yeah. For a cube, I think it is 6a squared. Okay, so a is the distance. I can't remember. I should have double checked this. I don't think volume is a cubed, so 2 times 2 times 2 or whatever. Okay, but then you would show it as, for example, if it was 64 to 32, you would need to show it as, well, 32 to 1, isn't it? Which is 64, which is 2 to 1, sorry. Okay, the way to do that is divide that into that, and then that's the 1, and that's whatever it's divided into.
In the situation where
Yeah, could be a decimal. Could be a decimal. Yeah. Good. Okay. So let's get on to gas exchange. So you need to know about gas exchange in, well, I could talk about gas exchange in mammals, but I'm not going to. I'm going to just concentrate on fish, plants, and insects today. Oh no, I suppose we need to mention mammals as well. Okay. Right. Right. Before I even talk about that, I'm going to talk about the principles. There are 3 main principles of gas exchange, and they are applicable to any example. All right, and I'd like you to think about these common things. So, number 1, principle number 1, we've got a large surface area. Okay, so for this is about gas exchange, exchanging gases. We're going to have a large surface area, usually. Secondly, what else are they going to have to allow diffusion to occur quickly? Thin membranes, good. So thin membranes. In fact, actually, I need to explain, don't I? So large surface area to increase the volume of gas exchanged. Thin membranes, what does that do to diffusion pathways? Yeah, shortens the diffusion pathway. Well done. So shortens diffusion pathway. Notice I'm giving a description and an explanation. And thirdly, for good gas exchange, you need to maintain a steep concentration gradient. So steep concentration gradient. What do I mean by that? I mean the concentrations are different between the exchange surfaces in and out of the exchange surface. Okay, so e.g. higher than lower concentration. So usually those are maintained by movement of blood or water, maintained by movement or circulation, movement of things that are circulated. Oh my God, that's terrible. I don't like that actually, so cross that out. So, I mean, in humans, that's blood circulation. So also it's ventilation as well. So in fish, ventilation is important. Okay, so movement of water across the gills, etc. Anyway, we'll talk about those. Right, let's talk about fish because they love fish. Hey, just as a, we got some new fish. I've got a 220 litre tank. Got phantoms, got tetras. It's great. I'm really pleased. Yeah. Anyway, so let's talk about fish gas exchange. Okay, anybody tell me something about fish, please? Okay, so you've gone immediately to countercurrent system. I'm going to go to their kind of circulation. So basically water goes through the mouth, through the gills. So the gills are the specialized surface. You need to know about gills. Okay, so gills. They're made of gill filaments. So gill filaments. Increase the surface area. And then those gill filaments are covered in lamellae. Okay, so you remember the gill filament, and then you've got the lamellae on it. Those have blood capillaries in them. It's not very good, that, oh well. Okay, so they're very thin, speed up diffusion. So I'm going to draw a little diagram. So here's oxygen concentration. Distance along the gill filament. So blood goes one way, okay? So blood goes that way. Blood, the other way is water. Now water has a low concentration of oxygen. I've got a blue. Yeah, so it goes the other way. So the key thing to know is the concentration of oxygen in the water is always higher than the blood. So the concentration of oxygen always higher blood across the whole length of the gill filament. Whole length gill filament. Okay. So that means diffusion is occurring right across, and this is known as you said, countercurrent multiplier. Where have you come across this as well before? You talk about it with kidneys, and the Looper Henley. Yeah. Okay, so that's fish. They can use mussels to pump the water in quicker, but I don't think you need to know about buccal cavities in AQA. Pretty sure of it. Oh. Okay. Right, let's talk about insects. So insects, they don't have, okay, so here's an insect. What do insects have? 1, 2, 3, 4, 6. They've got little holes or tracheoles, okay? So insects have got air-filled pipes called tracheoles or trachea, sorry. Yeah. So you've got trachea that go into tracheoles. Tracheoles. Those go to every cell in the body. So cells are close by. So it doesn't have any kind of circulation system, just diffusing it from outside. What there was a question, wasn't there, about abdominal pumping? Did you have to do that one? Abdominal pumping? No, that was your 12 progression test. So they can use muscles to contract and ventilate to move gases more quickly. What can they do to these tracheoles, this trachea? What can they do? Yeah, they can open and close them. So they can be open and closed. Open, close. You might be asked to calculate how long they're open or closed. So usually you see a graph like this. Like that. So when it's open is when oxygen concentration increases. So it's usually that point there. That's when they're open. You need to work out the time they're open. I like to do that. I'm not going to do plants. Oh no, actually, I will do plants. Let's talk about adaptations of water loss. Right, so obviously, plants, you should know about general plant structure, about controlling water loss as well. So they do want you to know about xerophytes. So xerophytes, things like marram grass. They can adapt to dry conditions. So things like stomata of sunken pits. So what I mean by that is that on the bottom of the leaf, kind of goes like this, and you get the guard cells there. Okay, so what happens is air comes along and just stays, water vapor stays there. So when it's windy, it doesn't get rid of all the water vapour. So that's wind. So it stops evapotranspiration occurring. They might have hairs on them as well, and layers of hairs. So it traps moisture in the stomata. Could have curled leaves, again to stop moisture leaving, and also less stomata as well. It's a real problem for plants of stomata because they need stomata for gas exchange, but they can lose lots of water vapour from them as well. Okay. Yeah. In gas exchange as well is about hemoglobin, about Bohr effect and things like that. I will just very briefly just talk about human lungs. So human lungs are dead easy. So lungs, you've got large, what gives it a large surface area? Yeah, millions of alveoli. What gives it a thin, what about the thin membranes? It's the alveoli and capillaries are 1 cell thick. And you've got ventilation that is maintaining a steep concentration gradient. There we go. You would also need to know about ventilation, but I'm not going to go through that because that's just a case of learning it. Oh, short, yeah, you need to mention as well short diffusion pathways. They do love short diffusion pathways. Good. Again, I mean, paper 1, there will be a bit of AO2. There might be a bit of looking at lung diseases and stuff like that. There is a bit of AO2, not as much as paper 2, okay, but there is a bit in there. Good, I think I'm going to stop there then. Okay, all right, so that's gas exchange in a nutshell. I hope you found that useful. And that concludes our video for today.